Hi, everyone. Welcome to the very first episode in YouthConnect's Research and Healthcare Interview Series. Hi, my name is Fatima Masood, and I'm a grade 12 student and a member of YouthConnect.
Hi, I'm Maya. I'm in grade 11 and I'm also part of the YouthConnect.
Hello, my name is Caroline. I'm a grade 12 student and I'm also a member of the YouthConnect.
So talking a little bit more about YouthConnect, YouthConnect was founded in late 2021 as a scientific outreach initiative for high school students, and we work towards introducing resources for those wanting to pursue a career in healthcare or in the field of research. We want to provide a platform and outlet for high school students to learn about research happening in their field of interest and to learn more from experts in those fields. And this webinar is a great way for students to do so. So for the first webinar episode, please join us in welcoming Dr. Chen from the Burn Research and Skin Regeneration Lab at Sunnybrook Research Institute in Toronto, Canada. So Dr. Chen is a research associate at the Sunnybrook Research Institute in Dr. Yeshke's research group, with a particular focus on developing stem cell therapies for burn patients. So Dr. Chen, would you like to introduce yourself as well?
Sure, thank you. Yeah, so my name is Yufei Chen. You can call me Andy. So as mentioned, I'm currently a research associate working under Dr. Mark Yeshke's lab. So basically, we're developing skin regeneration strategies for burn patients. And yeah, it's very glad that I can join you guys today.
Now, before we get into the research presentation, we would like to ask a quick round of questions to get to know Dr. Chen a bit better. The first question is, biology, chemistry, or physics? Which one is your favorite science subject and why?
So obviously I have to say biology. That's actually what I'm residing right now. So yeah, it's very exciting. I would say a major, if in the future you guys would consider going into lots of things that you can understand the human body or animals. So I would say it's very interesting.
Yeah, thanks for sharing that. And following the last question, what made you interested in entering your field of research?
So I would say definitely from the bottom of my heart that I want to develop something for healthcare system that can help our patient in a real life. So that's always the ultimate goal.
Lastly, what is one piece of advice that you would give to high school students who aspire to enter healthcare or research?
Yeah, so I would say utilize the resources around you. So try to participate in lots of research activities around you, especially go to maybe some workshop or laboratory at a university, understand what they're doing, so you can have a better understanding what's going on in current research and what's the future direction you want to move into.
Thank you for answering those questions. Now we can move on to the presentation where Dr. Chen shares his research.
Yeah, so I'll move on from here now. So thank you very much for this opportunity for me to talk about our research group at Dr. Mark Yeshke's lab. Mainly, I'll be focusing on the tissue engineering aspect of our research group. We're having a pretty large group at the Sunnybrook Hospital. And then tissue regeneration for skin is one of the major focus for our lab. So a little bit in background about epidemiology for thermal injuries. So as per WHO, a burn injury is an injury to the skin or other organs that usually caused by the hot liquids, solids, or even flames. And globally, it's over 180,000 deaths every year caused just by burn injuries. And then obviously large amounts of resources has been put on treating the patient. And that's a very heavy load for the healthcare system. And currently the standard of care is to generate a skin graft to cover the wound bed caused by the thermal injury. And usually the burned skin will be exercised first after the patient admission. And then the skin substitutes will be grafted on top of it to speed up the regeneration of the skin. So during the thermal injuries, there are multiple phases, starting from the initial insult of the thermal injury and during the healing of the skin. So that usually characterized with 4 different phases as shown in this slide here. First of all, it will start with the hemostasis. So basically, the fibrin will be clotting at the site of the wound, and then multiple cells, including fibroblasts or macrophages, will be recruited actually to the wound bed. And then that is followed by the local inflammation stage, which will be involved with lots of immune cells, including macrophages or neutrophils, filtrated at the local site to counteract the local infections. Obviously, after that is the skin regeneration phase. So we call it also the proliferation stage. That's involved multiple different type of cells, including macrophages, fibroblasts, endothelial cells. They're secreting multiple molecules, mainly it's the growth factors or cytokine chemokines to help regeneration of tissue, help increase the re-epithelialization to close the wounds locally. And after that is the final phase, which is the longest phase called remodeling phase. That includes multiple molecules to help the matrix remodeling to as close to as the healthy skin. And obviously, due to the severity of these thermal injuries, it will be characterized as different degrees of the injuries. So starting from the first degree, which is just superficial injury at the epidermis, and then have a deeper injury that we characterize the second or third degree. That's usually the insult will penetrate deeper through the epidermis, go into the dermis, or even completely destroy your dermis. So based on the literature, we actually have lots of patients that are suffering from either the second or third degrees of the thermal injuries. So before moving on to our research, I want to give a little bit introduction about the stem cells. So generally speaking, the stem cells are your body's own material that arose from different organs that helped the tissue to be regenerated on certain directions. And currently, based on the International Society for Cell and Gene Therapy, ISCT, they have defined criteria that a certain cell have to meet to be defined as the mesenchymal stem cells. So generally, they have to meet minimally 3 criteria as shown here. So first of all, they has to be adherent cells. They can adhere to a plastic vessel. So we usually in the lab, we culture the cells in the plastic vessel, either in a culture dish or flasks. So they have to be able to adhere to those plastic surfaces. And obviously, they have to be able to express certain surface antigen markers. So we call it cluster of differentiation markers, CDs. So if you look at the right panel over here, they have to be positive for CD 73, 90, 105, and also negative for certain CD molecules, such as CD 34 or CD 45. So all of this is actually will be characterized by a analytical methods called flow cytometry. So we'll stain those markers that express on the surface of the cells with certain fluorochrome. They show fluorescent signals, and then the flow cytometer can measure and detect those signals, and we can quantify how much of the signal has been expressing on those cells. And thirdly, obviously, the cells, the MSC cells, they have to show multipotent differentiation potential. So obviously, the minimum one that they have to show is to process the adipogenesis, osteogenesis, and chondrogenesis potential. And what we do is usually after isolation of the MSC cells, we culture them and then expose them to certain media, defined media, to guide their differentiation towards those 3 lineages. And then after usually 2 or 3 weeks, we stain with certain molecules and to show whether they are positive for those differentiation markers. So as I mentioned, during the burn care, once after admission of the burn patient, they are undergoing the first surgery, which is removal of their burned skin. So in our lab, we actually were able to exercise the burned skin and extract the mesenchymal stem cells from those burned skin. And those burned skin usually considered as medical waste. So extracting cells from it actually has the least ethical concern for the patients. And when we actually define those mesenchymal stem cells as burn-derived MSCs. So here is just a brief process how we isolate the cells from the burned skin. So obviously after the patient undergoing the first operation from the inside the operating room, and then we obtain their burned skin, and after that we receive the skin in the lab and remove any fat with the mechanical dissections, and after that wash it with ethanol and to help sterilize the tissue. And after that is multiple rounds of enzyme digestions to help dissociate the cells from the matrix of the skin. And after a couple of rounds of cell staining, which is to collect the cells from this digested matrix, we were able to pellet the cells and then later transfer into the flask for in vitro expansion. So to just briefly demonstrate the whole procedure for isolation. And after that, obviously, you have to characterize those cells. So basically, we have to show that you're able to express certain markers as defined by the ICT guidelines. So obviously, we're able to show that the burn-derived mesenchymal stem cells, they're expressing the three positive markers required for MSCs, including the CD73, 90, and 105. So as you can tell from the first paper that we published, and they were able to express at least 105 and CD73. And then later we perfect our isolation procedure, and we demonstrate that the PDMSCs can express all three markers at their passage, passage of 3 or 4. And then all of the PDMCs that we isolate demonstrate they were able to show tri-lineage differentiation as shown by the adipogenesis, chondrogenesis, and sterogenesis assay here. So they're showing positive signals basically after staining and exposed to the defined media after 2 or 3 weeks. Similarly, so we would later on, we compare those BDMSCs to the umbilical cord derived mesenchymal stem cells, because umbilical cord mesenchymal stem cells kind of like well-defined MSCs right now. So both of the MSCs demonstrate the positive expression of the markers and also able to show tri-lineage differentiation potential. And then we further compared the BDMSCs to UC MSCs in terms of their population doubling time. So that's actually a parameter to show how fast they can multiply themselves. So obviously there's no significant difference between these two types of MSCs. So they're quite similar. And colony formation is basically to show how many colonies they were able to generate during the in vitro culture, reflecting their proliferation potential. So again, very similar between UC MSCs and BDMSCs. And then we use a compound called BRDU to demonstrate their proliferation potential over a certain amount of time. And it's also very similar between those two. The reactive oxygen species are also quantified from the culture media for both of the cells to see whether they have a difference in terms of the secreted reactive oxygen species. So again, they're very similar. So overall, we can say the UC MSCs in vitro, after comparison, they're very similar. And then further on, we want to look at their immunogenicity aspect a little bit. So basically, that's actually defined by their surface marker expression of the major histocompatibility complex molecules, so which is the MHC1 and MHC2 molecules. Even though we have a smaller size of the sample, but both of the MSCs demonstrate quite low expression of both MHC1 and MHC2 type molecules, and they are not very significant difference. So kind of similar, which shows the potential that they will pose very little amount of immunogenicity to the host. And then we look at a whole bunch of different secretory molecules from these two types of MSCs. So again, without the stimulation using a certain compound, which is, if you can look at the lighter in color of the bars. So if you look at, for example, the, I would say the IL-6, which is a cytokine that's secreted from the both of the MSCs, they're quite similar without the stimulation. So which is their basal line. And among all of the tested secretory molecules, none of them actually showed very significant differences in terms of between these two MSCs. So that's also give us an idea that their secretory profile is probably very similar. So which gives us the confidence that the BD-MSCs may demonstrate very similar regeneration potential compared to the umbilical cord derived MSCs. So obviously, before we moving on to test their efficacy, we want to look at their safety profiles, which usually is a great concern for any cell or gene therapy product. So first of all, let's look at the tumorigenicity aspect of the MSCs. So on the left panel here, you can appreciate that which there's no significant of the cell growth after we implant the cells, inject the cells actually to a nude mice, to the back of the nude mice. So we, in this study, we try to inject the BD UCMSCs, also the fibroblast and a cancer cell line to the nude mice to see if there's any tumor formation after, I think it's 20 days. So obviously in vitro, we didn't see a large amount of cell cluster formation post 20 days. And then that's corresponding to the in vivo observations as well. We didn't see a tumor generation or formation post 20 days from any of the MSCs or fibroblasts. But for cancer cells, we obviously see a tumor actually formation after 20 days. So this also demonstrates the safety using a small animal. Our BD-MSC is not tumorigenesis. And then again, this is also the confirmation about the flow cytometry analysis on the MHC class, MHC molecules. They're having a very low expression levels. So that's also a side proof to support the low tumorigenicity issue for the host. And then later I'll move on to look at their efficacy after transplanting the BD-MSCs to the nude mice. And then we look at over a course of 12 days and see how good the wound will be healed after we injected the BD-MSCs to the local wound. In this case, it's an excisional wound caused by a puncture. So it's a round puncture that has been created on the skin of the nude mice. And then after 12 days, we definitely see a closure of the wound bed. And if you look at the histo images over here, the arrow demonstrates the cross-sectional aspects of the wound, and the shorter length of the wound has been observed when the wound bed was treated with the BDMSs comparing to without the BDMSs. So that indicates a quicker closure of the wound due to the regeneration, probably assisted by the BDMSs. And also after we quantify the average wound size, that's also significant lower comparing to the control if the pneumomice was treated with the BDMSs. So that's a promising result arising from the small animals. And then later on, we move on to our established large animals using the pig. So in our lab, we developed this protocol that we can introduce a severe thermal injuries to a large animal like a pig. And the pig will receive this injury just caused by a heated metal that is actually 5 by 5 in centimeters in the squared shape. So after the pig was treated by this metal, so we heat it up to 200 degrees Celsius. And then we actually seeded the BDMSs on the carrier material. It's called Integra dermal transplant. So this is an industrial collaborator that we're working with. It's a skin scaffold, but without any cells. So we seeded the BDMSs onto those Integra, which we called it acellular Integra containing no cells. And we compared that with the Integra that has been seeded with BDMSs. So we monitored the wound for over a course of 1 month and look at the closure of the wound, their growth images over the time, and look at their reepithelialization rate. So if you look at the panel A over here, the arrow indicates the front line of the new epidermis, which we utilize that to calculate the reepithelialization rate. So if you compare to the acellular Integra, we definitely have like large amount of newly formed epithelia. And then after we quantify that reepithelialized area, the BDMS treated group is actually more than the acellular group. And then if you look at, the different vascular or blood vessel formation, the BDMS treated one also demonstrate more blood vessel. If you look as demonstrated, shown in a darker circle over here. So that's actually the CD31 positive blood vessel that's stained. And then if you quantify that, obviously it's a higher amount of vascularization comparing to the one without BDMSs. And then further on, we want to look at if there is a dose responsive effect on MSCs for our thermal injury pig model. So we firstly try to look at the UCMSCs on pig instead of burn-derived. So the whole procedure is very similar. So we isolate the UCMSCs from human Walden's jelly. That's actually from the human umbilical cord. In vitro expand them, and then we did the FACS, which is the flow cytometry analysis, to sort out the UCMSCs. And after that is to seed them onto this dermal regeneration template from Integra. We have two groups, same as the previous study. So either acellular control or it was seeded with MSCs. And after that, we grafted them onto the burn model we have for the pig and monitor them for overall 4 weeks. So if you look at the growth images in the panel B on day 28, we actually observed a lower dosage of the cells actually have a better healing in terms of the wound closure. So the 200 or 5K cells per square centimeters demonstrate a better wound closure, so smaller wound that's still open. And if you compare it to either the higher dosage or a acellular control or burn alone, it's definitely much smaller. And the cause of that, it could be the more cells that we seeded onto the integra just deplete the nutrition surrounding them, and then that's actually hindered their efficacy. And then after that, we definitely want to zoom in to the local histological analysis among those groups. So basically, when you look at the right panel over here, we have 4 different groups. So basically, our control group is the burn alone without any treatment, which is this group. And then the control group is actually the acellular. We also call it material control. So basically, those group of pigs receive only the integra material without any cells seeded. And then obviously we have the experimental group that has the integra with the MSC seeded onto it. And obviously we compare all 3 groups to the healthy skin of the pig as a positive, as actually a negative control group. So we actually demonstrate different aspects in terms of histological analysis. So first we will look at the morphology epidermis regeneration. So from the if you look at the healthy skin, they have obviously the red stain for the epidermis, and then dermal compartment, there's a large amount of collagen. Once you do the trichrome staining, they will be stained as blue. So that's the darker the blue signal indicate the high degrees intensity of the collagen. So basically, if you look at the morphology of epidermis, healthy skins like that, and then the burned one, they completely lost their epidermis. And then the control group, which is the acellular control group, no cells incorporated, they have some epidermis restored, but obviously with the cells, we have much better restored epidermis. And then if you look at the dermis, so that's mainly characterized by the intensity of the blue signal. So obviously healthy skin has the strongest. And then the rest of 3 groups, the treated one with the cells demonstrate much higher signal in terms of blue. And then next thing is to moving on, we look at the vascularization post treatment. So healthy skin have multiple blood vessels embedded in their dermis. So burn alone restores some, and then our cell-containing one also restore quite a bit. And then the other aspect to look at whether the regenerative skin is good or bad is based on the fibrosis of the local tissue. So the lower degree of fibrosis indicate a better healing outcome. So by looking at that, we stain a positive marker to indicate the fibrosis, which is alpha SMA. And obviously, if it's a healthy skin, you have much lower level of the alpha SMA. And then without any treatment, just burn, you have a whole bunch of increased alpha SMA expression. But with the cell-treated one, we were able to decrease that amount quite significantly. And then the other 2 markers, CD11B and CD163, is for pro-inflammation. So definitely the lower level, the better. So obviously, the cell-containing group actually have a much better outcome comparing to either without the treatment or acellular control group. So generally speaking, now we can see that the MSCs that isolated from the burn skin have a efficient regeneration for skin when we seed it onto a material that is integra. And also the low dose of the MSCs demonstrate much better skin regeneration. And then the other aspect our group also interested in is these extracellular vehicles called exosome that's usually secreted from the mesenchymal stem cells. So what we found is actually we were able to identify those exosomes from the Walden's jelly, human Walden's jelly of that's actually from as part of the human umbilical cord. We were able to isolate those exosomes from the umbilical cord tissue, and then what we did is actually to image them to show whether they will be absorbed by the fibroblast. So these two images over here, the darker is actually the SEM images that demonstrate the darker dots, which is the exosome that can be observed by the fibroblast. And then after we have the exosome isolated, we actually try to treat the fibroblast with the isolated exosome. Either they're not disrupted, which is still intact, so we called it whole exosome, or they were lysed to expose any materials that they carried with inside. And then we actually observed after 24 hours, the intact exosome has the best outcome in terms of the cell migration. So this assay is called a in vitro scratch assay. So what we do is we see the fibroblast in the well plate, usually it's a 6-well plate, and then we introduce the artificial scratch by using a pipette tip, so onto a confluent monolayer of the cell, and that is actually as shown in blank over here, so you don't see any cells in between. And then we switch the media to either the media containing an intact exosome or lysed exosome for over 24 hours and see if the cells will migrate from these two sides towards the blank space in between. So the more the cells are present after 24 hours, definitely demonstrate that better migrations potential of the media. So obviously the intact exosome has the best outcome. So then we would ask the question, what's causing this? And we're interested in what's the main driving force or molecule help this. So what we did is we analyzed the overall protein profile by a technique called proteomics. So basically you extract the total protein from the intact exosome, and then that will be analyzed by a technique called mass spectrometry to actually quantify it. What's the molecule, the proteins or peptide present in your sample. So what we found is actually the alpha-2 microglobulin has quite a bit of expression or level in the lysate. And then based on that, we treated, again, using a scratch assay, treated the fibroblast with 2 dosage of the alpha-2 macroglobulin, and to see whether that will help the migration of the cells. And so obviously it did. And then the higher the concentration that we utilized, then the increase the migration we can observe after 24 hours. So this is also one other aspect of our research we are interested in, and we currently are continuing this direction to see if we can also identify similar effects from the exosome secreted by the PDMSEs as well. So, and then further on, we want to look at if there is a dose responsive effect on MSCs for our thermal injury peak model. So we firstly try to look at the UCMSEs on pig instead of burn derived. So the whole procedure is very similar. So we isolate the UCMSEs from human Walden's jelly. That's actually from the human umbilical cord. In vitro expand them, and then we did the FACS, which is the flow cytometry analysis, to sort out the UCMSEs, and after that is to seed them onto this dermal regeneration template from Integra, We have two groups, same as the previous study. So either acellular control or it was seeded with MSCs. And after that, we grafted them onto the burn model we have for the pig and monitor them for overall 4 weeks. So if you look at the gross images in the panel B on day 28, we actually observed a lower dosage of the cells can actually have a better healing in terms of the wound closure. So the 200 or 5K cells per square centimeters demonstrate a better wound closure, so smaller wound that's still open. And if you compare it to either the higher dosage or a acellular control or burn alone, it's definitely much smaller. And the cause of that, it could be the more cells that we seeded onto the integra just deplete the nutrition surrounding them, and then that's actually hindered their efficacy. And then after that, we definitely want to zoom into the local histological analysis among those groups. So basically, when you look at the right panel over here, we have 4 different groups. So basically, our control group is the burn alone without any treatment, which is this group. And then the control group is actually the acellular. We also call it material control. So basically, those group of pigs receive only the integra material without any cells seeded. And then obviously we have the experimental group that has the integra with the MSC seeded onto it. And obviously we compare all 3 groups to the healthy skin of the pig as a positive, as actually a negative control group. So we actually demonstrate different aspects in terms of histological analysis. So first we will look at the morphology epidermis regeneration. So from the if you look at the healthy skin, they have obviously the red stain for the epidermis, and then dermal compartment, there's a large amount of collagen. Once you do the trichrome staining, they will be stained as blue. So that's the darker the blue signal indicate the high degrees intensity of the collagen. So basically, if you look at the morphology of your epidermis, healthy skins like that, and then the burned one, they completely lost their epidermis. And then the control group, which is the acellular control group, no cells incorporated, they have some epidermis restored, but obviously with the cells, we have much better restored epidermis. And then if you look at the dermis, so that's mainly characterized by the intensity of the blue signal. So obviously healthy skin has the strongest. And then the rest of 3 groups, the treated one with the cells demonstrate much higher signal in terms of blue. And then next thing is to moving on, we look at the vascularization post treatment. So healthy skin have multiple blood vessels embedded in their dermis. So burn alone restores some, and then our cell-containing one also restore quite a bit. And then the other aspect to look at whether the regenerative skin is good or bad is based on the fibrosis of the local tissue. So the lower degree of fibrosis indicate a better healing outcome. So by looking at that, we stain a positive marker to indicate the fibrosis, which is alpha SMA. And obviously, if it's a healthy skin, you have much lower level of the alpha SMA. And then without any treatment, just burn, you have a whole bunch of increased alpha SMA expression. But with the cell-treated one, we were able to decrease that amount quite significantly. And then the other 2 markers, CD11B and CD163, is for pro-inflammation. So definitely the lower level, the better. So obviously, the cell-containing group actually have a much better outcome comparing to either without the treatment or acellular control group. So generally speaking, now we can see that the MSCs that isolated from the burn skin have a efficient regeneration for skin when we seed it onto a material that is Integra. And also the low dose of the MSCs demonstrated much better skin regeneration. And then the other aspect our group also interested in is these extracellular vehicles called exosome that's usually secreted from the mesenchymal stem cells. So what we found is actually we were able to identify those exosomes from the Walden's jelly, human Walden's jelly of that's actually from as part of the human umbilical cord. We were able to isolate those exosomes from the umbilical cord tissue. And then what we did is actually to image them to show whether they will be absorbed by the fibroblast. So these two images over here, the darker is actually the SEM images that demonstrate the darker dots, which is the exosome that can be observed by the fibroblast. And then after we have the exosome isolated, we actually try to treat the fibroblast with the isolated exosome. Either they're not disrupted, which is still intact, so we called it whole exosome, or they were lysed to expose any materials that they carried with inside. And then we actually observed after 24 hours, the intact exosome has the best outcome in terms of the cell migration. So this assay is called a in vitro scratch assay. So what we do is we see the fibroblast in the well plate, usually it's a 6-well plate, and then we introduce the artificial scratch by using a pipette tip, so onto a confluent monolayer of the cell, and that is actually as shown in blank over here, so you don't see any cells in between. And then we switch the media to either the media containing an intact exosome or lysed exosome for over 24 hours and see if the cells will migrate from these two sides towards the blank space in between. So the more the cells are present after 24 hours, definitely demonstrate that better migrations potential of the media. So obviously the intact exosome has the best outcome. So then we would ask the question, what's causing this? And we're interested in what's the main driving force or molecule help this. So what we did is we analyzed the overall protein profile by a technique called proteomics. So basically you extract over the total protein from the intact exosome, and then that will be analyzed by a technique called mass spectrometry to actually quantify what's the molecule, the proteins or peptide present in your sample. So what we found is actually the alpha-2 microglobulin has quite a bit of expression or level in the lysate. And then based on that, we treated, again, using a scratch assay, treated the fibroblast with 2 dosage of the alpha-2 macroglobulin, and to see whether that will help the migration of the cells. And so obviously it did. And then the higher the concentration that we utilized, then the increase of migration we can observe after 24 hours. So this is also one other aspect of our research we are interested in, and we currently are continuing this direction to see if we can also identify similar effects from the exosomes secreted by the PDMSEs as well. So yeah, so overall, I would like to give this to you guys about our research, specifically focusing on skin regeneration based on mesenchymal stem cells that we developed or isolated from human organ. And yeah, I would definitely like to acknowledge our funding agencies that's shown here and University of Toronto and Sunnybrook Hospital. So yeah, if you guys have any questions, I'm happy to take them.
Thank you, Dr. Chen, for sharing with us an insightful introduction to your research. Now, could we ask a few specific questions surrounding your research and the healthcare field? To start with, what educational or career training did you take to become a scientist and researcher?
So basically, I started my major in bachelor. That's actually in the pharmaceutical engineering. And then we were exposed to lots of different aspects of biology. So that's encompassing from cell biology, molecular biology, immunology, and also core aspect of pharmaceutical sciences, including pharmaceutical analysis, pharmacology, pharmaceutical chemistry. So that's given me I think, the fundamental view about how the biochemistry or cellular biology will help to treat or prevent certain diseases. And then after that, I joined University of Manitoba College of Pharmacy, basically focusing on studying a molecule called liver fatty acid binding proteins. That's actually for treating fat liver diseases. And then after that, I accomplished my PhD degree in the pharmaceutical sciences, mainly focusing on developing medical devices or prophylaxis for HIV or AIDS infections. And then after that, I joined a local company here in Canada, the biopharmaceutical company, specialized on fabricating biopharmaceutical agents or drugs. And that exposed me to a lot of, I would say, GMP-based manufacturing processes, and also the regulatory aspect of approving those investigational product for human use. Later on, I would like to come back to academia just mainly for continuing my interest in pharmaceutical sciences and tissue engineering. So that actually landed me in my current position as the postdoc, and then later on as research associate working in Dr. Mark Yeshke's lab.
Thank you so much. And moving on to the next question. If implemented, how is this research anticipated to improve patient experience when recovering from a burn?
That's a great question. So as I've shown here, the greatest, I think, discovery from this project is actually we were able to isolate the burn-derived mesenchymal stem cells from the medical waste, usually considered as medical waste, the burn skin from patient. So that gave us a great opportunity that we can develop a autologous stem cell therapy that we can isolate patients' own stem cells from their skin tissue with little or no ethical consideration issues, and then develop a stem cell therapy to turn that into a skin substitutes and then graft on the same patient. And that will actually open a door, I think, for the future burn care, that we can hopefully increase or enhance the skin regeneration with much less host rejection. So currently we already obtained Health Canada approval to start this trial, so which is very exciting. It's a first in human phase 1 trial. So I'm looking forward that we can start recruiting our patient very soon. Yeah.
Well, thank you so much for that answer. And that's really exciting to hear that it's been approved. So along the same lines of that, of your answer, what was the initial motivation for this clinical trial to use autologous cells to treat burns, which normally would be discarded?
Yeah, so I think based on the current standard of care, there's lots of issues. First of all, it's not a lot of grafting material that's available. So usually, ideally would be if we can have at least host rejection, we have to harvest the healthy skin from the same patient. We call this donor site. That's actually from the same patient, but the healthy skin area. But the issue is that usually when we admit a patient, they were actually suffering from large burn, end up like you don't have a lot of space for you to harvest that donor site from. And then the other issue is like if you harvest that, then it's another injury to the patient as well. So to actually prevent that, I think this is definitely one of the advantage that we can use the cells from the patient, but we say different skin substitutes that without causing a third or a second insult to the patient, and also under the circumstances that we don't have the donor site available. I think that's actually the beauty of this treatment, and also due to based on our in vivo and in vitro preclinical study results so far, we can see the much like greater enhancement in terms of skin regeneration can be observed using those cells. So that could be a great, I think, treatment for the patient in the future.
Well, yes, I think that's so it's like, so the beauty of science is like having the ability to continue to research and like have these clinical trials to further our knowledge. So I guess you kind of touched on this as well, but I guess like to have its own question, like what path do you envision your research and practice taking in the future? So you kind of touched on this in the last one, but if you want to sort of expand a little bit further.
Sure. Yeah. So I think we discovered the BDMSCs 2018, and then there's so much more that is not answered for the cells. So we have to understand every aspect of the cells and their potential to be developed into a cell therapy. And also, I think the material delivery strategy for the cells is also matters so much. That's also we have currently collaboration with other groups at University of Toronto or other industrial collaborators to incorporate those cells and then to develop better or easy to use or clinical compatible material delivery strategy incorporating the cells. So that's also why I think that's also the next step that we have to move into. Yeah, and then other aspect, once we pass the phase 1 trial, if we can demonstrate it's safe, then obviously it's to look at their efficacy, like how many cells we have to use. Obviously, there's a certain amount of cells we can isolate from it, but we have to show or have to understand what's the optimal dose of the cells we can use to help the patient. So that will be, I think, be answered in the phase 2 of the study.
Dr. Chen, thank you so much for this opportunity. It was really nice to hear, like, for us high school student, and I'm sure like every high school student would like benefit from this experience. Thank you for joining us today, Dr. Chen. And this presentation has been very informative and has taught us a lot about skin regeneration and field of skin regeneration in general. YouthConnect will be posting more webinars in the near future, so please stay tuned on our social medias and check out our blog page for more future information. Also, stay tuned for our summer healthcare conference. Our social media will be in the description along with Dr. Chen's contact information, so feel free to contact him with your questions related to his research. Thank you all for listening, and thank you, Dr. Chen, once again.
Thank you.
Bye.
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 Yeah.